So, good afternoon everybody and thank you for coming. Today I'll be talking about my PhD presentation. So, the objectives of my project is to investigate whether genetic variation within of risk genes is associated with cognitive decline. And in particular, I'll be looking at single nucleotide polymorphisms and genetic risk scores, which is a combination of the SNPs. And also determine whether there's interactions between these genetic risk scores and environmental factors. So why do we want to look at cognitive declining? Well, due to improvements in medicine and healthcare, the world's population is beginning to age. An individual can now expect to spend a quarter of their life over the age of 65. This is going to result in 2050 of 2 billion people being aged over 60 worldwide. So with aging, there's a general loss of function, both physical and cognitive, that has an impact on a person's abilities to perform everyday activities. So in terms of cognition, a normal rate of cognitive decline is considered to be about 6% decline in general cognitive ability per decade. However, it's important to recognise that not all cognitive domains actually decline over time. You can see that in the first graph on this slide, where domains associated with crystallised intelligence such as vocabulary and general knowledge, these actually tend to improve at a rate of 0.02 to 0.03 standard deviations per year. Conversely, domains associated with fluid intelligence, so memory, perceptual speed, executive functioning, these actually are the domains that tend to decline. And again, you can see that in the graph where domains associated with fluid intelligence are declining from at the age of 20 to 90. However, the rate of cognitive decline is very heterogeneous within the population, which you can see in this graph. Some people are actually staying relatively stable in their cognitive abilities over time, while others are declining at a much more rapid rate. So it's important to understand what causes this heterogeneity within the population. And this is important because with normal cognitive aging, this has an impact on a person's ability to, as I said earlier, to perform everyday tasks. And this is important because contemporary life requires an individual to use their cognitive abilities throughout every day. This is from simple things just as filling out forms and working out public transportation schedules to more complex matters such as household finance and medication. So longitudinal studies have found there is an association between the rate of cognitive decline and a person's ability to perform essential everyday tasks. And this naturally results in a reduced quality of life and overall life satisfaction for those individuals who have a greater rate of decline. However, for individuals that have a more accelerated rate of decline, they often transition into dementia, of which Alzheimer's disease is the most common. So Alzheimer's is characterized by impairment of multiple cognitive capacities. That results in them being unable to live independently and requiring a caregiver, and this then often results in death. However, it's difficult to distinguish between pathological decline and normal cognitive aging, which you can see in these two graphs here. So in the first graph, you can see that the preclinical stages of Alzheimer's disease, which can occur up to 15 years before onset dementia, has a considerable overlap with normal cognitive aging. And within the second graph, you can see this in terms of the biomarkers associated with Alzheimer's disease, where the first four lines are corresponding to amyloid beta accumulation, synapse dysfunction, tau-mediated neuronal injury, and brain structure. And abnormal levels of these, or abnormal characteristics of these biomarkers, can occur fairly on in the preclinical stage of dementia, before you start to see any cognitive deficits, which is the second last line. This pattern is also observed at the end of an individual's lifespan, where autopsies of non-demented individuals have indicated they often have neuropathological changes that are the same as those associated with Alzheimer's, yet, as I said, these people don't have any cognitive deficits. So, in understanding some of these sources, or some of the sources of variation for this heterogeneity in cognitive aging, one of these is of course going to be genetic. And there's been a number of approaches that have been utilised to investigate potential genes that could be associated with cognition. So one approach is to look at late onset Alzheimer's disease genes. So since 2000 there's been about 10 genes that have been identified as associated with late onset Alzheimer's disease. So it's possible, since you've got such overlap between the preclinical stages of Alzheimer's and cognitive aging, it's possible that these genes could also influence cognition. And there's also the assumption that since the single load risk factors have small effect sizes in terms of Alzheimer's disease, it's possible that by themselves they're not going to cause Alzheimer's but cause cognitive loss. So this approach has been met with mixed success, with conflicting reports for individual genes. Although ApoE, which is the largest genetic risk factor for Alzheimer's, actually has been fairly consistently associated with Alzheimer's, especially in the most recent studies. So another approach is to look at neurotransmitters, as these form the basis of cognitive functioning. And this has identified several promising candidates, but the results for the majority of these remain fairly inconclusive. You can also look at endophenotypes, as they represent a more quantifiable and measure that occupies a space between genetics and the disease phenotype you're looking at. And two of the most important endophenotypes to look at is neuroanatomical differences. So this is because general brain size is highly associated with cognitive performance. And with aging, you tend to see atrophy in the hippocampus. So genes that are associated with neuroanatomical difference could also be associated with cognition. And also you've got cardiovascular disease, so hypertension increases small vessel disease, and this results in cerebrovascular lesions. And this is particularly apparent in vascular dementia. So again, genes associated with cardiovascular disease could also be associated with cognition. An alternative approach to looking at these candidate gene studies is to utilize a genome-wide association study. So this is a hypothesis-free approach that identifies whether common genetic variations, so single nucleotide polymorphisms, are associated with the phenotype of interest, so in this case cognitive decline. However, this approach requires very large sample sizes, so this has complicated this approach in terms of replicating some findings that genome-wide association studies have found. So over the last 17 years, we basically haven't had much luck in identifying genes that are highly associated with cognitive decline. We have found numerous putative risk genes, but most of these haven't been replicated, apart from ApoE, as I said earlier. At least those have indicated that 24% of the variation in cognitive decline is due to genetics. So a lot more investigation needs to be done on putative risk genes that have been found. So the reason why all these initial studies haven't been replicated is because of the nature of cognitive decline. So it's a highly polygenic trait with lots of genes and small effect sizes, but also because of the characteristics of the studies that have investigated genetic risk factors. So they tend to have inadequate sample sizes, suffer from issues of population stratification. There's confounding results due to environmental exposure and also variation in how cognitive decline has been classified and measured. So genetics isn't the only source of variation for cognitive decline. You're also going to get environmental and lifestyle factors. So this is increased risk associated with cognitive decline has been found for cardiovascular disease, poor overall health, diabetes, metabolism, metabolic syndromes, poor diet and tobacco use. While decreased risk has been associated with physical activity, cognitive training, healthy diet, moderate alcohol consumption, education. Other putative risk factors have also been proposed, but again, most of these remain somewhat inconclusive. However, we're also beginning to realize that the genetic and environmental lifestyle factors just don't operate in isolation of each other. There's interactions between them. So this is where genetic effects can be modulated by environmental variability, or genes can modify sensitivity to environmental factors. So one particular genetic and lifestyle factor that interacts with each other is hypertension and ApoE. So hypertension has been increasingly linked with dementia and cognitive decline, and there's evidence for is mainly associated with midlife hypertension rather than late life hypertension, which is somewhat mixed. So it's hypothesized that high blood pressure induces atherosclerotic conditions within the brain, and this alters cerebral blood flow autoregulation, and this promotes clinical and subclinical brain damage that then results in neurodegeneration. APOE, as I said earlier, is a large genetic risk factor for Alzheimer's disease, and it's also associated with non-pathological aging. So it's hypothesized that the different alleles of APOE, so E2, E3, and E4, have differential binding to amyloid beta peptides within the brain, and this results in differential regulation of amyloid beta aggregation and clearance in the brain, and that leads to synaptic dysfunction and neurodegeneration. So this is the predominant hypothesis. There have been other hypotheses that are independent of amyloid beta that have been proposed, but this tends to be the most predominant one. So several longitudinal studies have investigated the effect of a hypertension ApoE interaction on cognition, but the results from these studies again remain inconclusive. And this is mainly due to differences in participant characteristics in each study, so the age, education, ethnicity, and also the number of participants you have in your study, and also the methodologies that studies have used. So this tends to be how cognition has been measured, the measurements of blood pressure, how long the study's gone for, so how many years and how many time points they have, and this also then affects the statistical methods they can use to identify association. So this led to the first research question I was looking at in my PhD. So this was to elucidate whether the ApoE genotype moderates the relationship between late life hypertension and cognition. And for this I'll be using data collected from the Personality and Total Health, or Path Through Life cohort, which is a longitudinal study of community-dwelling individuals that is investigating the well-being of these individuals over time, with a particular focus on depression, anxiety, substance abuse, and cognitive ability. So these individuals were selected from the Canberra and Queanbeyan region, and the individuals were recruited into 3 separate cohorts based on the age of recruitment, so the 20s, the 40s, and the 60s cohorts. And data is collected from these individuals at 4-year intervals. And so far we have 3 waves of data, so this is for 8 years. There's currently a 4th wave of data being collected. So for this study I'm primarily interested in the 60s cohort, which has a mean age of 62.5 years old, education of about 14 years, and 51% of them are male. So the first step in identifying whether there's an association between the hypertension and ApoE interaction with cognitive decline is actually to remove individuals who may bias your results. So this included individuals who only attended 1 wave, those who had a medical history for issues such as brain tumours and probable dementia, and also individuals who weren't Caucasian, and also to exclude individuals if they had missing values for some of the variables they're looking at, such as ApoE genotype and smoking. Alternatively, you can impute missing values, which is what I did for education and blood pressure variables I had. I did this using expectation maximization. So this left me with a total sample size of about 1,700 people. So in terms of the variables I'll be looking at, my predictors are going to be ApoE genotypes. This is just E4 carriers versus E4 non-carriers. I'll also, of course, be looking at hypertension. So this is just based off your systolic over diastolic blood pressure, or if you're taking hypertensive medication. And then just a continuous measure of mean arterial blood pressure, which is the mean arterial pressure variable. My outcome variables then will be the neurocognitive tests associated with the path data. So this is looking at perceptual speed, short-term memory, episodic memory, working memory, and vocabulary. And then also a global cognition score based off the average of all these. In addition, I have a measure of some motor skills, which is the Purdue pegboard. My covariates then will be the demographic variables I have in the study, so gender, smoking, education, heart trouble, diabetes, and depression. So to identify associations, I'm using a statistical method called multi-level modeling. So this models change over time to quantify the relationship between a continuous dependent variable and various predictor variables. So multi-level modeling is characterized by its nested data structure it has. So this is where your first level is nested within your second level. And in terms of longitudinal studies, this is repeated measurements over time nested within your subject. It's also characterized by having random and fixed effects in the model. So the fixed effects are how averaging across the population or the individuals, your populations and subpopulations change over time. So essentially this is just the intercepts and slopes. Your random effects then are the variance components, and they represent how much variation there is away from the fixed x. This can be used to determine the amount of variance explained at each level of the model. So when you put the path into a schematic that sort of represents multi-level modeling, you get the schematic up on the slide here. So at our level 1 we have our time variables, so this is age, and our neurocognitive tests. Well, at level 2 we have our subject-specific variables, so our predictive variables ApoE, hypertension, and mean arterial pressure, and our covariates. So altogether, this is going to allow us to investigate the within individual change, so how individual outcome values change over time. This is describing the relationship of each person's individual growth trajectory. Then we can also look at the individual differences in change. So what predicts within individual change and describes the relationship. And this describes the relationship between the predictors and the shape of each person's individual growth trajectory. So multi-level modeling also involves the construction of multiple models. So the first model you have is your null model, or the unconditional means model. And this is characterized by not having any predictors or time variables in it. And this allows us to establish baseline values for establishing model fit and variance explained, which we'll come to shortly. Your next model then is your unconditional growth model, where time is introduced. And this allows us to calculate the average rate of change within the population. So you then introduce your covariates in the covariate model, and this controls the effect that these covariates may have on cognition. You then introduce your predictor variables, so ApoE, hypertension, and mean arterial pressure. This allows us to establish the effect that the predictor variables themselves have on the rate of change. Then in our final model, the interaction model, we can establish whether the interaction affects the rate of change and explains any additional variance. And this is just introducing the interaction term between the blood pressure variables and ApoE. So since we're constructing multiple models, we actually have to determine which model best explains the data that we're looking at. So this is the model selection step, and this is simply just involves using likelihood ratio tests, and this is determined by subtracting the -2 likelihood of the reference model from the nested model, and then determining significance by referring to a chi-square distribution. We can also look at pseudo R-squared statistics to determine the amount of variation explained in each model as well. So this involves, so once you run all these models through the software, this creates tables and tables worth of data. So I won't show you every single one, but I will show you specifically the global cognition hypertension model. So you can see that in our first model, the unconditional means model, as I said earlier, this is just allowing us to establish the baseline values for variance explained and the model fit, which you see down at the bottom of the slide. So in our second model then we're introducing time, and when we do this you can see that there is a significant decline in global cognition scores over time, and that the time variable is improving the model fit and explaining an additional 0.7% of the variance. So in model 3 where we introduce our covariates, again we can see that when we do this the model fit is improving, and also the amount of variation explained is increasing to 18%. So in our next model we're introducing the ApoE and hypertension terms. You can see that when we do this, the ApoE E4 is associated with a further decline in global cognition scores over time. This is significant. However, when we look at the change in model fit, this has not been improved, and the amount of variance explained is only 0.1%. Then when we look at the interaction model between, so the interaction term between ApoE and hypertension, we can see that this interaction term further results in a decline in global cognition scores. However, again, when we look at the change in model fit, which is in red, we can see that this is not improved. And furthermore, the amount of variance explained is only 0. So when we looked at the other neurocognitive tests, we saw a similar pattern in the results. So the only cognitive test that had a significant effect for the interaction term was spot the word, global cognition, which I just showed you, and purge pegboard dominant hand. However, when we looked at the model fit, only spot the word had an improvement. But when we looked at the additional variance, that was explained by adding the interaction terms, it was negligible for all the cognitive measures. And furthermore, the results was insignificant after correcting for multiple testing using Bonferroni correction. And this was the same for the mean arterial pressure models as well. So all up, these results seem to indicate that, well, there is a significant association between the ApoE hypertension interaction term and cognition, that due to the improvements in model fit and additional variance explained being negligible, it's most likely that ApoE doesn't actually modulate the effect late life hypertension has on cognition. So these results are similar to those of two previous studies by Caselli and Notman, who utilised a very similar methodology to what we did. And also they had looked at a similar number of subjects. We had slightly more, but they still had around 1,000. And also they had similar measures of cognition. However, their results and ours contradict those of the two most recent studies by Bangin and Yasuno. He did find an association between hypertension and ApoE with language ability and global cognition. However, they utilised a different statistical method to what we and Caselli and Notman utilised. So the PATH cohort, however, has several advantages over these previous studies, notably that it has it's investigating a large number of cognitive tests, 2 measures of blood pressure, so hypertension and mean arterial pressure. There's a larger number of individuals in the cohort. And also, especially in comparison to Bangin and Yasuno, we have multiple time points in our data, which allows us to use the multi-level model to look at our change over time, which they couldn't do because they only had 2 time points. And the statistical methods that they use are not as robust for looking at a change over time. However, it should be noted that we were looking at a late life hypertension, and that if you utilised a measure of midlife hypertension, you may identify a more robust association. This is, as I said earlier during the introduction, that midlife hypertension has been more strongly associated with cognition than late life hypertension. Also, if you looked at individuals who had dementia rather than non-pathological decline, again you may see a more robust association. And this is because in dementia there tends to be overlap between Alzheimer's, which is generally characterized by the ApoE allele, and vascular dementia, which is characterized by neurocardiovascular disease. However, there are a couple of limitations associated with our cohort. First of all, that's relatively well educated, so this generally results in a higher cognitive reserve in these individuals, which is associated with a less decline in cognition over time. And also our population is a little bit too young at the moment, so the studies that have generally found a strong association between ApoE and cognition have had a slightly older population in the mid-70s, while ours are just into the early 70s now. But with once we get a new wave of data in, it's possible that we'll see a more stronger association in our cohort, because they'll be slightly older. So ApoE isn't the only gene that's going to be associated with cognition. As I said earlier, a number of putative risk genes have been identified. It's just that the majority of these haven't been replicated, and the evidence for these remains inconclusive. So the second research question associated with my PhD project is to identify whether other putative genetic risk factors are associated with cognitive decline in the PATH cohort. So as I said earlier, there's been a number of genes that have been proposed to be a risk factor for cognitive decline, and these have been associated with low-risk genes, neurotransmitters, and phenotypes, or variants identified through genome-wide association studies. So I'm particularly interested in the genes that have been previously associated with late-onset Alzheimer's disease, which you can see in this table here. So ApoE has actually been previously genotyped by our group, but I'll be looking particularly at the most recently identified risk genes for Alzheimer's. And these have been associated with pathways to do with lipid metabolism like ApoE, also immune system and synaptic mechanisms. So we selected 64 SNPs that had been previously associated with their dementia, cognition, neuroanatomical differences of blood pressure, and we genotyped these 64 SNPs within the PATH cohort. So that's about 4,500 individuals from the 40s and the 60s cohorts. So our genotyping, or to genotype our samples, we utilised the TACMAN OpenRay system by Biosystems. Initially this involved a preamplification, so this is because the path DNA samples aren't characterized or normalized and the DNA concentrations are relatively low. So this reduces the absolute template copy number you have available, which can then affect the quality of the data obtained from the TACMAN reactions. So we just performed a multiplex PCR amplification of the targeted loci to essentially boost the template copy number available. So the actual genotyping was then done using TACMAN open array reactions, and this is just a real-time PCR step that generates some fluorescent signals that can then be used to determine allelic discrimination. So you can actually see in the diagram here what one of the open array plates looks like. So you can see that each square represents a sample, so 48 samples per chip, and then each square has 64 dots in it, which represent assays for the SNPs you're looking at. So the actual genotype calling was performed in the TACMAN genotyper software, and I just used automatic calling automatic calling, and each cohort was called separately. So after I had genotyped these samples, I had to perform a number of quality control checks to ensure our data was the quality is robust and good to use. So initially I looked at identifying samples that had identical genotype calls between the two samples, and this was to identify sources of pipetting error. So to do this, I just performed a pairwise alignment to identify individuals with 90% similarity in their genotype calls. And this identified 17 pairs of duplicates initially, and after these had been repeated, this was reduced to 2 pairs. You can see visually what these duplicates look like in the samples I have on the screen here. So you can see the primary, if you look at the genotype calls, so the patterns of the colours essentially in each square, you can see that the primary samples are exactly the same as the duplicate sample there. So a subset of the path cohort of 620 individuals actually been previously genotyped for SNPs associated with Compton, BDNF. So it's possible for me to compare the genotype calls I had for these samples to those that had been previously obtained and see whether there was any discordance between the two data sets. Initially it was found that there was 8 samples that had a call rate, genotype calls that were different. After these had been repeated, this was reduced to 3. You can see where these 3 samples are located within their respective genotype clusters. So on the left you have my calls from the open array. You can see there's one individual in each cluster there. However, when we looked at the genotype calls for the original compt calls, which you can see on the right, these same 3 individuals should be called as non-amplified. However, I should note this is after I ran it through the genotyper software, which is a different software to what was originally used. So this indicates that these 3 original calls may have been of rather low quality to begin with. So a third quality control check I used was using the identifying heterozygous haploids within Plink, the Plink software. And this is because we had 2 X-linked SNPs, so there shouldn't be any heterozygous males in the heterozygote clusters. However, we initially identified 10 samples that were males, heterozygotes for these 2 SNPs. However, after these have been repeated, this was reduced to 5 samples, so 1 in the 60s and 4 in the 40s cohorts. So in terms of call rates, there was only 9 out of the 4,500 samples that had a call rate less than 90%. For individual SNPs, there's only 1 in the 40s cohort that had a call rate less than 95%, which you can see on the graph up here. So if you look at the graph on the left, you can see there was a large number of individuals who were called as undetermined because they fell in between the homozygote and the heterozygote clusters. So this is what would cause the lower call rate for this SNP. So in terms of Hardy-Weinberg equilibrium, I performed a Fisher's exact test within Plink to identify SNPs that were out of Hardy-Weinberg. And this identified initially 6 SNPs within the 40s cohort and another 6 SNPs in the 60s cohort that were out of Hardy-Weinberg. However, this was at a p-value cutoff of 0.05. Once you correct for multiple testing, this only leaves 2 SNPs, PARP1 and TRIM65, that are out of Hardy-Weinberg equilibrium. So these quality control checks indicated that the data was fairly robust and it's good to use for the data analysis stage now. So as I said earlier, initially I'm primarily interested in the genes that have been previously associated with late onset Alzheimer's disease, so these are the ones up on the screen here. So initially I'll be looking at whether the individual SNPs are associated with cognitive decline, but also be looking at whether there's an interaction between the most recently identified SNPs and APOE. However, if you look at the odds ratios here, you can see that for the most recently identified ones, they actually are rather small. So it's possible that we won't see an association between these individual SNPs and cognition. So an alternative method to analysing this data is actually to create genetic risk scores. And there's several ways this can be calculated. So the first way is using a simple count measure. This is just adding up the number of risk alleles each individual has. So this is good when you don't know the effect size for each SNP with the phenotype you're looking at, and we don't actually know what this is for cognition, although we do know what it is for late onset Alzheimer's disease, which is the odds ratios I have up on the board. However, this measure makes the presumption that each SNP is contributing equally to the phenotype you're investigating. As you can see by looking at the odds ratios for our SNPs, that this is not the case, especially for APOE. So what you can then do is weight each individual risk allele based on the odds ratios, which is the second method of calculating a genetic risk score. A third method which has been recently proposed is to look at the explained variance, and this is weighting each allele not just by the odds ratio but by the minor allele frequency, which is also on the screen here as well. So this is based on the assumption that minor allele frequency and the odds ratio are important for determining genetic heritability and variance. So I'll be testing whether these individual SNPs or the genetic risk scores are associated with cognitive decline by using multi-level modelling, pretty much exactly the same way that I use multi-level modelling for the APOE hypertension reactions I showed you earlier. And also, once I looked at these individual SNPs and genetic risk scores, I'll also start introducing environmental and lifestyle factors into the equations as well, to see whether there's any interactions between them. So, in conclusion, this project initially looked at identifying whether there was a gene and environmental interaction. Particularly I was looking at whether the APOE genotype moderates the effect of hypertension or cognitive decline. And analysis of the data from the 6-path cohort suggested that this was not the case. So my second research question I'll be looking at, which I'll be doing for pretty well the rest of this year, is investigating whether genetic variation with imputated risk genes is associated with cognitive decline. So to do this, I've genotyped 64 SNPs within 4,500 individuals across the 40s and 60s cohorts from the PATH dataset, and the data analysis for this stage is ongoing. So thank you for listening. I'd just like to thank my supervisors, Debjani, Simon and Karen, who have provided me with lots of advice and the opportunity to work on this project. Also Jorge Velez, who has helped me a lot with writing R scripts and doing a lot of the quality control checks. And also Susan Tan, who's provided me a lot of help in the lab, and also other members of our lab from the Genome Diversity and Health Group. Anyway, thank you for listening.